Most beer lovers know the bittersweet taste of hops. Aroma hops are very flavorful, but require many resources to produce, including large quantities of carbon dioxide and thousands of liters of water for just 1 kilogram. Producing the same flavors by using genetically modified yeast would save considerable money and resources. A group of researchers in Denmark has achieved this. Yeast starts to produce flavors by creating geranyl diphosphate, a universal building block for many of the flavors in hops. The yeast naturally produces geranyl diphosphate from a substance that is plentiful in yeast cells, acetyl coenzyme A, acetyl-CoA for short. This happens because the cytoplasm of the yeast cell has several enzymes that convert acetyl-CoA step by step to geranyl diphosphate. Such a chain reaction is called a biosynthetic pathway, and living cells have many of these pathways. A biochemical sports commentator would probably explain a biosynthetic pathway like this. We're live at the mevalonate pathway, and today's lineup: on one side is acetyl-CoA, and on the other all sorts of cytoplasmic enzymes, and we're very excited to see whether the enzymes will hit their target by converting acetyl-CoA to geranyl diphosphate. Now the contest has really started, with acetyl-CoA colliding with a thiolase, and thiolase binding with one more acetyl-CoA, creating an even larger molecule, acetoacetyl-CoA, which immediately collides with the next enzyme, a synthase, that reacts strongly by binding to yet another acetyl-CoA while absorbing water and creating an even larger molecule, hydroxymethyl-glutaryl-CoA. Wait, here comes a reductase, which uses NADH to reduce the carbonyl group to a hydroxy group and creating mevalonate. Yes, ladies and gentlemen, mevalonate, the molecule that gives its name to the whole pathway. Whoa, suddenly mevalonate collides with kinase, the enzyme that uses ATP to attach phosphate groups to molecules. So now we have mevalonate 5-phosphate. But here comes one more kinase and attaches another phosphate group. And this runs right into a decarboxylase, which can remove carboxylic acid groups. And yes, the carboxylic acid group has been removed, simply detached and released as carbon dioxide. And now we have isopentenyl diphosphate. Yes, this has a carbon-carbon double bond at the end of the carbon chain, but hang on. Here comes an isomerase, the enzyme that can rearrange molecules internally to form another isomer of the molecule. Now the double bond is suddenly further inside the chain, and dimethyl allyl diphosphate is formed, and this reaction just continues in a ping-pong process. Now, here comes the last enzyme, natural ERG20P, and it can attack two molecules at the same time. It takes on both isopentenyl diphosphate and dimethyl allyl diphosphate, and the enzyme wins. Geranyl diphosphate has now been formed, and the enzymes are victorious. Yes, that is how the synthesis plays out. But there is a problem. The synthesis pathway is not quite finished. The natural ERG20P can create geranyl diphosphate, but it can and will also convert geranyl diphosphate into a new substance, farnesyl diphosphate. This last step snatches the valuable geranyl diphosphate from the researchers who wanted to use it to make hop flavors. What can they do? The research group in Denmark had a good idea. They genetically modified the yeast by inserting special versions of the genes encoding for all the enzymes in the mevalonate pathway.
First, we inserted a special version of ERG20 protein, which can create geranyl diphosphate but does not convert it into farnesyl diphosphate. Second, we modified all the enzymes by adding some extra amino acids in one end that ensure that they are located in a separate organelle in the cell, called the peroxisome.
The native enzyme, which would have otherwise removed geranyl diphosphate, cannot penetrate the peroxisomes, so now the yeast cell can produce geranyl diphosphate unhindered, which can be further converted into various hop flavors. Thus, the yeast cells can now replace the resource-intensive production of hops.
Compared with using hops, the new technology can reduce water consumption by 10,000 times and carbon dioxide emissions by 100 times for the same amount of beer. And even though the yeast is genetically modified, the products are not, and are completely identical to the natural flavors.
So, if we want to make our lives more sustainable and reduce greenhouse gas emissions, this is a much more sustainable way to produce hop flavors. You can read more about this research at sciencenews.dk.